All right, another deep dive. Welcome. And today we're going to try to dig into serotonin receptors.
Oh, very cool.
You know, those things that actually allow serotonin to, do stuff.
Yeah.
It's not just about mood, though.
Right.
It's digestion, sleep.
Oh, wow.
Even your heart needs serotonin to do its thing.
And that's what's so fascinating about these receptors, I think.
Yeah.
Serotonin could have so many different effects because you have multiple types of serotonin receptors. Each one has its own special role.
So not just one receptor that's calling all the shots.
Right.
Makes sense when you think about how many different processes serotonin's involved in.
Exactly. Exactly.
Yeah.
And to make it even more interesting,
Okay.
they fall into two main families.
Okay.
GPCRs, or G protein-coupled receptors.
I remember those from
Yeah,
biochem.
ion channels.
Yeah, those were the complex signaling cascades, right?
That's right.
Like a chain reaction inside the cell.
It sets off this whole cascade of events, and it ultimately changes how the cell behaves.
Right.
Ion channels, on the other hand, are more direct.
Okay.
Those are like gates that control the flow of ions
Okay.
across the cell membrane,
Makes sense.
and that can quickly change the electrical activity of a cell.
Gotcha. So two very different mechanisms.
Yes.
Both triggered by serotonin.
Uh-huh.
But
Yes.
let's focus on those GPCRs.
Okay.
For a sec, you mentioned there's a lot of diversity within that family.
There is, yeah.
Okay.
We have subtypes like 5-HT1,
Okay.
5-HT2, and so on.
Yeah.
Each subtype has its own personality,
Oh.
you could say, and they influence different pathways and functions within the cell.
So it's like having different keys that fit into the same lock.
Yeah,
But each one unlocks a slightly different room.
that's a great analogy, yeah.
Okay.
And here's where things get really interesting.
Okay.
You can have different serotonin receptors,
Uh-huh.
even ones from the same subtype on the same cell.
Yep.
And even if they activate the same signaling pathways, they can have completely distinct effects.
Wait, hold on. How is that possible?
Yeah.
They're triggering the same pathway.
Right.
Shouldn't the outcome be the same?
Right.
It's like pressing the same button but getting different results.
That's where the idea of microdomains comes in.
Microdomains.
Or signaling compartments.
Okay.
Think of it this way.
Okay.
Imagine a factory with all these different departments,
Okay.
and each one specializes in a particular task,
Gotcha.
even though they might all use the same raw materials.
Okay.
The final products they produce are distinct.
I see the parallel.
Yeah.
So different serotonin receptors might be localized to different
Sure.
microdomains within the cell,
Exactly.
even if they activate the same pathways.
Yeah. And this challenges the old way of thinking about cell signaling.
Okay.
We used to think that signaling molecules would just diffuse evenly throughout the cell,
Right. A global signal.
like turning on all the lights in the house. But microdomains suggest that things are way more localized.
Right. So like turning on only the lights in a particular room.
Yeah, precisely.
Gotcha.
So to illustrate this,
Oh.
let's look at cardiomyocytes,
Okay.
the cells that make up your heart muscle.
Yeah.
They have a variety of serotonin receptors, as
Okay.
well as other receptors
Right.
that activate the same signaling pathway,
Okay.
which involves this molecule called cyclic AMP.
So stimulating any of those receptors should increase cyclic AMP levels.
Right.
But you said the effects can be different.
Exactly. So let's say you stimulate the beta-1 adrenergic receptor
Okay.
on a cardiomyocyte.
Okay.
This leads to an increase in cyclic AMP,
Mhm.
which ultimately causes the heart muscle to contract with more force.
So like adrenaline gets your heart pumping.
Right, exactly.
Yeah.
But now let's say you stimulate a different receptor
Okay.
on the same cell, the EP4 receptor.
Okay.
This also leads to an increase in cyclic AMP, but it doesn't affect the heart muscle's contractility at all.
Whoa.
Same pathway, different outcome.
So the same pathway can have opposite effects depending on which receptor kicks it off
Exactly.
and in which microdomain it's happening.
Yeah.
Like having two light switches
Uh-huh.
that both control the lights, but one dims them and the other brightens them.
Exactly.
Wow, that's mind-blowing.
And that's why understanding this microdomain organization is so important,
Okay.
especially when we talk about things like drug development.
Okay, I'm starting to see the connection.
Yeah.
If different drugs target different receptors,
Right.
even within the same family,
Yeah.
they could have very specific effects depending on which microdomains those receptors are located in.
Precisely.
Wow.
And to make these microdomains work,
Yeah.
the cell relies on special scaffolding proteins
Okay.
called A-kinase anchoring proteins, or AKPs.
AKPs, those sound familiar.
Yeah.
Are they like the organizers of the cell, keeping everything in its place?
You could say that, yeah.
Okay.
They act like architects designing and building those distinct microdomains.
Gotcha.
They bind to a key enzyme
Okay.
and the cyclic AMP pathway called protein kinase A, or PKA,
Hmm.
and anchor it to specific locations within the cell.
So it's like assigning PKA to different workstations within the factory,
Yeah,
ensuring that each step of the manufacturing process happens in the right place.
that's a great way to visualize it.
I try.
Yeah, and each AKA has a specific targeting domain
Okay.
that determines where it and its associated PKA will be located within the cell.
So different AKPs, different locations, different PKA pools.
Exactly.
That makes sense. But I'm curious, how does PKA actually get anchored to the AKP?
Right.
Is there like a specific mechanism involved?
There is, yeah. It involves a special structure
Okay.
called an amphipathic helix,
Okay.
which is part of the AKP's PKA binding domain.
Gotcha.
This helix specifically interacts with a certain type of PKA called type 2 PKA,
Okay.
which is the type that's typically found in specific subcellular locations.
So it's like a lock and key mechanism.
Yes.
Ensuring that the right type of PKA is anchored to the right AKE in the right location.
Yes.
A lot of specificity.
Exactly. And this intricate system of AKPs and PKA anchoring
Yeah.
is what allows for such precise control over signaling within the cell.
Okay.
Different receptors can activate different local pools of PKA,
Uh-huh.
leading to distinct downstream responses, even if they're activating the same pathway.
This is really highlighting the complexity of cell signaling, isn't it?
It is.
It's not just about the receptors themselves, it's about their location,
Yes.
their interactions with other proteins,
Uh-huh.
the specific pathways they activate.
Absolutely. And this complexity has huge implications for how we understand the effects of medications,
Okay.
especially drugs that target specific serotonin receptors like SSRIs.
Right, the antidepressants.
Yeah.
We talked about those earlier.
Yeah.
They work by increasing serotonin levels in the brain.
Yeah.
But now I'm starting to see how this microdomain organization
Yeah.
could play a role in their effect.
Exactly. Different people might respond differently to SSRIs
Okay.
because of variations in their receptor subtypes, aka P profiles,
Okay.
or the organization of their microdomains.
Mhm.
It's personalized medicine at the cellular level.
Okay, so it's not just about flooding the brain with serotonin.
Right.
It's about understanding the nuanced dance of its receptors
Exactly.
and how they orchestrate such diverse effects throughout the body.
Yes.
But before we get too deep into SSRIs,
Okay.
I'm curious to learn more about the different serotonin receptor subtypes themselves.
Sure.
We've touched on the 5-HT1 and 5-HT2 families, but what are some of the key players within those groups?
Yeah.
And how do their roles differ?
That's a great question. That's a great question. It's like exploring different neighborhoods within a city, you know?
Okay,
Each one has its own unique character and function.
gotcha.
Let's start with the 5-HT1 family,
Okay.
which is known for its generally inhibitory effects, often leading to a decrease in neuronal activity.
So if serotonin is like a messenger delivering instructions,
Yeah.
the 5-HT1 receptors are telling the cell to slow down or like take a break?
That's a good way to think about it.
Uh,
They often act like the brakes in the nervous system.
Mhm.
And within this family, you have subtypes like 5-HT1A,
Okay.
which is highly concentrated in brain regions associated with mood and anxiety.
So that's why it's a target for some antidepressants and anti-anxiety medications.
Exactly.
Gotcha.
Now, in contrast to the 5-HT1 family,
Yeah.
we have the 5-HT2 family,
Okay.
which is more often associated with excitatory effects.
So they're hitting the gas pedal instead of the brakes.
Exactly.
Okay.
And within this family, you've got subtypes like 5-HT2A,
Okay.
which plays a role in a wide range of processes, from perception and learning
Hmm.
to the actions of certain psychedelic drugs.
Wow, so these receptors are involved in some pretty profound experiences.
They are.
It's amazing to think that these tiny molecules are orchestrating such a complex symphony of effects within our brains.
Yeah, it is really amazing.
What about that 5-HT3 receptor we mentioned earlier?
Right.
The one that's an ion channel.
Right. The 5-HT3 receptor is unique because it doesn't rely on those intricate signaling cascades like the GPCRs do.
Okay.
Instead, when serotonin binds to it, it directly opens a channel
Oh,
that allows ions to flow across the cell membrane,
uh-huh.
leading to a rapid change in the cell's electrical activity.
So it's like flipping a switch instead of sending a message through a chain of command.
Exactly. And this rapid action makes 5-HT3 receptors particularly important in processes
Okay.
that require quick responses,
Okay.
like nausea and vomiting,
Make sense.
as well as pain perception.
You mentioned earlier that serotonin is also involved in digestion.
Yeah,
Do these different receptor subtypes play a role in that too?
absolutely. The interplay between these different subtypes is crucial
Okay.
for regulating gut motility. The movement of food through your digestive system.
So it's like a carefully choreographed dance between
Uh-huh.
excitation and inhibition,
Yes.
all thanks to these various serotonin receptors working together.
You got it. And that's just within the serotonin system itself.
Right.
When you consider the potential for synergy with other neurotransmitters like dopamine and norepinephrine,
Yeah.
the complexity just explodes exponentially.
Wow.
It's like adding more instruments to the orchestra,
Yeah.
creating a richer and more dynamic symphony of effects.
It's amazing how all these systems are interconnected, influencing each other to create the tapestry of our experiences.
Yeah, it really is remarkable.
But let's zoom back in on those serotonin receptors for a second.
Okay.
We've talked about their diversity and their roles in different processes,
Uh-huh.
but I'm curious about the pathways themselves.
Okay.
What happens downstream when these receptors are activated?
Right.
We touched on cyclic AMP earlier, but what are the actual steps involved?
That's where things get even more intricate. It's like opening up a watch and seeing all the tiny gears and springs working together.
I like that analogy.
So imagine a serotonin molecule
Okay.
binding to a 5-HT receptor
Okay.
that's coupled to a G protein called GAS.
GS, is that like a special type of G protein?
It stands for stimulatory G protein.
Okay.
And its job is to activate an enzyme called adenyl cyclase.
And adenyl cyclase is the enzyme that produces cyclic AMP.
Right.
So it's
Yeah.
like the first domino in the cascade.
Exactly. Once adenyl cyclase is activated,
Okay.
it converts ATP, the cell's energy currency,
Okay.
into cyclic AMP.
Okay, so cyclic AMP levels start to rise within the cell.
Right.
What happens next?
Well, cyclic AMP then goes on to activate another key enzyme
Okay.
called protein kinase A, or PKA.
Right, the enzyme that gets anchored to specific locations within the cell.
Exactly.
So cyclic AMP is like the messenger that activates PKA,
Ah,
sending it off to do its job.
precisely. And once PKA is activated,
Hi.
it can phosphorylate a wide range of target proteins.
Phosphorylate.
It basically attaches a phosphate group to other proteins,
Okay.
which can change their activity, their shape,
Okay.
or even their location within the cell.
So it's like adding a sticky note to a file,
Yeah.
telling the cell to pay attention to this one or send this one over there.
That's a great way to put it.
Okay.
And the specific targets of PKA can vary
Ah.
depending on the cell type and the context,
Okay.
which is why the same serotonin-activated pathway can lead to such diverse outcomes.
Okay, so it's like a chain reaction of molecular events.
Yes.
Each step amplifying the signal
Right.
and leading to a more widespread impact within the cell.
Exactly.
But wait, you mentioned that there are other pathways involved in serotonin signaling,
Yes.
not just the CAMP pathway.
Right.
What are some of those?
Well, another important one is the phosphoinositide pathway,
Okay.
or PI pathway for short.
Gotcha.
This one involves a different set of second messengers,
Okay.
molecules that relay the signal from the receptor to other parts of the cell.
So instead of cyclic AMP, the PI pathway uses different messengers.
Right.
What are they and how do they work?
The key players in the PI pathway
Right.
are IP3,
Okay.
or inositol triphosphate, and DAG, or diacylglycerol.
Those are some complicated names.
They are, but their roles are fascinating.
Okay.
IP3 acts like a key that unlocks calcium stores within the cell.
Okay.
It binds to receptors on a structure called the endoplasmic reticulum,
Uh-huh.
causing it to release calcium ions into the cytoplasm.
Calcium, I know that's an important signaling molecule, but I'm not sure exactly why.
Yeah, calcium is like a universal signaling currency
Okay.
within the cell.
Okay.
It's involved in a wide range of processes, from muscle contraction to neurotransmitter release.
Okay.
So when IP3 triggers the release of calcium,
Uh-huh.
it's like setting off a chain reaction of events
Okay.
that can have a huge impact on cell behavior.
So IP3 is like a conductor setting off a wave of calcium signaling within the cell.
Yes, that's a good way to put it.
What about DAG? What does it do?
DAG stays within the cell membrane
Okay.
and activates an enzyme called protein kinase C, or PKC.
Another kinase.
Yes.
So both the KMP pathway and the PI pathway ultimately lead to the activation of kinases,
That's right.
which then go on to phosphorylate other proteins.
Exactly. And PKC, like PKA, can phosphorylate a wide range of targets, influencing everything from gene expression
Okay.
to cell growth. and differentiation.
So it's all about controlling which proteins are active
Yes.
and where they're located within the cell.
Exactly.
This is truly mind-boggling.
It is.
It's like a whole hidden universe of signaling complexity happening right beneath our noses.
And we're just beginning to scratch the surface.
Wow.
The more we delve into these pathways, the more we realize how intricate and beautifully orchestrated they are.
It's a reminder that even the simplest actions, like feeling happy or experiencing a gut feeling, are the result of this intricate dance of molecules and signaling pathways.
Precisely. And understanding these complexities is not just an academic exercise.
Okay.
It's crucial for developing treatments that target these pathways with precision, addressing the root causes of diseases rather than just masking the symptoms.
Wow, this has been like a fascinating deep dive.
It has.
We've gone from the basics of serotonin to the intricacies of receptor subtypes and microdomains. These like crazy interconnected signaling pathways.
It's a lot.
And it sounds like we're just scratching the surface.
Yeah, we really are.
It's a vast and complex field.
Absolutely.
But I guess the more we understand about these signaling networks, the better equipped we'll be to develop treatments that target them with precision and effectiveness.
Exactly.
Speaking of treatments, you mentioned SSRIs earlier.
Yes.
I'm starting to see how understanding all of this complexity could help us develop better medications, especially for things like depression and anxiety.
That's right. Remember, SSRIs work by increasing serotonin levels in the brain. But they don't just boost serotonin everywhere. They primarily target the 5-HT1A receptor subtype.
The one that's concentrated in brain regions associated with mood and anxiety.
Exactly.
So by increasing serotonin's availability at those specific receptors, SSRIs can help regulate mood and reduce anxiety.
Exactly. And the fact that SSRIs target specific receptor subtypes highlights why understanding this complexity is so crucial.
It's not just about serotonin itself. It's about understanding the nuance of its receptors. And how they orchestrate such specific effects throughout the body.
It's amazing.
This brings us back to personalized medicine.
It does.
Doesn't it?
Yeah.
If we can identify which receptor subtypes or pathways are dysregulated in a particular individual, we could potentially develop treatments that are more precise and effective.
Exactly.
With fewer side effects.
That's the goal.
It's like having a toolbox full of different tools, each designed for a specific purpose.
Yeah, like that.
By understanding which tools are needed for each individual, we can tailor our treatments to achieve the best possible outcome.
Absolutely.
That makes me really hopeful for the future of mental health treatment.
Me too.
Imagine a world where we can target these pathways with such precision that we can address the root causes of these conditions.
Exactly.
Not just the symptoms.
I love it.
This whole conversation's been a real eye-opener for me.
Good.
I used to think of serotonin as this simple feel-good molecule. But now I see it as a key player in this vast and intricate network of signaling pathways that orchestrate so many aspects of our lives.
It's really remarkable.
It's a humbling realization.
It is.
And it makes you appreciate the incredible power of science to unlock these secrets.
For sure.
Well, on that note, I want to thank you for joining us on this incredible deep dive.
It's been a pleasure.
Into the world of serotonin receptors.
Fascinating stuff.
It's clear we've only just scratched the surface. But hopefully you've gained a newfound appreciation.
I hope so.
For the complexity and beauty of these systems.
Yes, and perhaps a little bit of awe.
Absolutely.
At the incredible dance of molecules that makes life possible.
Thanks for listening.